altered the gene, so this has fed into the public fear that genome editing is not going to stop it relieving diseases. It's going to end up in cosmetic alterations to the human genome, once again feeding into designer babies. Mice have also been used as models for human disease. They've used mice to test genomic deletions that in humans lead to disease, and they were able to then have 37 of the pups no longer possess these mutations and these diseases, but once again 50% did exhibit mosaicism. This does show that much more research is needed on CRISPR technology before it's used in humans clinically, as we need to minimize these risks before we go start testing on people. Although we have performed a human study, so they injected the Cas9 with the sperm into the egg because they believed this would help minimize mosaicism in a human embryo. 72.7% of the injected embryos were able to develop to the 8-cell stage, and out of these 50% were able to develop to blastocysts. They were then tested the entire genome looking for off-target mutations, and the results showed very few, showing that CRISPR-Cas9 does have strong specificity in humans. This would be then able to increase the amount of viable embryos for IVF treatments for couples that are experiencing infertility. There is an alternative to CRISPR-Cas9. This is a different strain of Cas9 that comes from Staphylococcus aureus, which is this bacteria. This strain of Cas9 is much smaller with a unique binding site, which is thought to increase the efficiency of the Cas9 because it will have less to bind to, so it won't have to find a long strain of DNA to bind to. So in comparing the two different strains of Cas9, we can see that the cleavage rates appear to be vastly different, with the Staphylococcus aureus, which is the one over here, having about an 87% cleavage rate, meaning 87% of the embryos tested were able to break the DNA to be changed, versus over here only having about 49% of the embryos being able to be changed. The birth rate for the Staphylococcus aureus was around 19%, whereas the typically used Cas9 was closer to 10, which is very similar. And then as far as the rate of mutants out of the embryos that were able to develop to full term and be born, for Staphylococcus aureus Cas9 being once again closer to 90, with the typical Cas9 being closer to 40. So this would make it appear that the Staphylococcus aureus Cas9 has a much higher efficiency, but according to the study, when actually compared with every different gene they've tested, they actually have much more similar efficiencies than this would depict. So I don't know how that exactly is working. It must be very gene dependent. So we should definitely look into using both of these as technologies instead of just focusing on one. So we do need a lot of different future studies in order to make gene editing functional in a clinical setting, including an increase in animal models. We need more animal models, as many that have been performed had very small sample sizes, that's the word. They've had very small sample sizes with a lot of negative outcomes of mosaicism and off-target mutagenesis. So if we increase the amount of studies we're doing before we go to humans, we can ensure that it's going to be safe for clinical use. We should also look into using mitochondrial transfer so that we can take a mother with damaged or mutated mitochondria, and we can remove the nucleus out of the egg, take a donor woman with healthy mitochondria and remove the nucleus, and combine the two. And then once it is fertilized, it will have healthy mitochondria, so the child will then have 3 parents' DNA. Unfortunately, this will only work for disorders that are caused by mutations in mitochondrial DNA. It won't be able to fix everything, but this will allow us to fix certain disorders without the use of CRISPR, so we'll be able to avoid all of the negative effects such as off-target mutagenesis. We should also look into more into precise genome editing. So being able to take a specific section of DNA and take it out and replace it, as this is used a lot in plants, and if we were able to transfer the technologies used in plants to being used in animals, and then people will have a much larger tool belt for treating human disease.